Hello. In this part of the MOOC, we'll talk about the case when it's difficult to find experimental values for your model parameters. So you have a problem of under-constrained parameters, and we would like to use parameter optimization, in particular meta-heuristics such as evolutionary algorithms, to help us building good neuron models. When you want the computer to help you with that, you have to be able to guide the computer what is a good model and what is a bad model. And for that, you'll need some way of assessing the goodness of fit of your model. Furthermore, we will try to make a case that using multi-objective optimization is a good ingredient to sort of expose certain trade-offs in the parameter space. And we'll give you an example that hopefully convinces you that these are useful techniques to help us build detailed neuron models. And of course, as always, there are some caveats you will have to consider. Let's get started. In previous parts of the MOOC, we were talking about and framing the problem by saying you have a detailed neuron morphology and you have certain electrophysiological recordings that you want to express in mathematical terms so that you have a computational model of a neuron that then can recreate this behavior. We showed you that there are different data sets that you should consider when building up these models. It starts with morphology. Ideally, you have a list and identification of ion channels that are creating the electrical behavior. Possibly you have kinetic information about how these channels behave. And ideally, you have also information where these channels are and how much of a certain channel is in a certain part of the dendrites. As we were saying earlier, sort of some of these data sets might be easier to get for the neuron of interest, and particular other data sets, like, for example, the localization and the maximum conductance, might be less available. So when we're talking about resorting to computer-based search techniques, of course, in principle, you could apply them to any parameter in this space, but in particular, we'll talk about applying them to finding the right location of a channel and the maximum conductance. So what to do with under-constrained parameters? Of course, the natural course of science is that you would hope that there will be more experiments, maybe even novel types of experiments that can measure aspects that were previously not measurable. And then, of course, you simply wait until this data becomes available, and then you use that data. Now, if you can't wait that long, or essentially it is unlikely that this will be done, you can, of course, resort to guessing these parameters and hand-tune them. You can be systematic about and simply sample the different parameters in a regular fashion, but that might become very easily a large problem, especially if you have many parameters to search for. Another strategy altogether could be to say, well, maybe these are not all independent parameters, but maybe you can express some relation between two parameters, for example, the conductance at two different locations of the dendrite, and instead of searching for them as individual parameters, maybe you can express this relation in a function and sort of that has less parameters in the first place. A yet other way of thinking about it is that maybe you can infer parameters from other and more easily measurable properties. So take the example of the ion channel conductance. Ultimately, what it means is out of how many channels are in a certain unit area of the dendrite. Now, typically you could sort of say, maybe I can count them under a microscope, but if you can't do that, why not infer at least a maximum bound of that value by saying, looking at the size of the ion channel complex that it occupies in a surface, and then calculate how much surface you have in this part of the dendrite altogether. So at least you could infer how many channels maximally could fit into a certain patch of dendrite. So while this is not giving you the answer of the direct parameter, it at least gives you an upper value, which can be very useful. So in this spirit, of course, you could imagine other ways of inferring properties. But if all that doesn't help you, then one possibility is to resort to parameter optimization. Parameter optimization is a mathematical field which you can express in mathematical terms that, for example, for a function f of x, which this might be the function, you're looking for a certain x0 that fulfills a certain criterion. For example, that the evaluation of a function at that x0 is the minimum for all of x. So you're looking for example, the global minimum of a function. This is a long-standing problem in mathematics, and different, a large variety of methods have been developed for solving these problems. And first and foremost, of course, you have calculus-based methods where you can simply say, okay, if I'm somewhere in this function, I possibly can calculate the derivative and then sort of, for example, descend to a local minimum. Or I can maybe do the second derivative and find all my extremer positions and so on. So there's, if your function is continuous and has certain properties, you can use these calculus-based methods. Another group of methods is when you can express your problem in different terms. For example, if you can express your problem as a set of linear equations and then sort of express the constraints in the same way, and then you can use a technique called linear programming to address the optimization in those cases. And then more generally, there's another class of methods which fall under the term of iterative methods that do less assumptions about the properties of the function. And this is useful because in our case, as a matter of fact, we don't want to make particular assumptions about our function. What is our function? We'll come to that. But ultimately, this is the neuron model you want to evaluate, and it might be not fulfilling properties that are required for calculus-based methods or linear programming. And of course, in all of that is you want to do this with a minimum number of function evaluations, because ultimately, this is what will cost you the time. If you have to sort of calculate the function at every x, this might take a long time. A variant of parameter optimization is, instead of trying to set out to find the global minimum, global maximum, you might be able to get away with finding a sufficiently good solution to an optimization problem. And that may be because you have incomplete information about the problem in the first place and limited compute capacity, limited time at your disposal. So a meta heuristic says, instead of looking for this global optimum, maybe you can sort of have an approximation or a set of reasonably good solutions. And in that sense, the meta heuristics is not a categorization of a calculus-based method or an iterative method, but in fact it can be a combination of the two, and people have done and combined, for example, local gradient descent methods with global heuristics such as simulated annealing. And a variant of these meta heuristics are population-based meta heuristics, like, for example, swarm optimization or evolutionary algorithms, where instead of sort of having one search point, you have a multitude of search points and treating them at the same time. The good thing about meta heuristics is that they make very few assumptions on the optimization problem, and essentially, therefore, can also give you approximate solutions to otherwise possibly intractable problems. But that comes with the downside of it, that essentially they don't give you a guarantee on whether this is a globally optimal solution, and they tightly and closely depend on the random number quality you use for your sampling of the space. So here you just need to be wary that running your search algorithm with different random seeds and good random numbers is a good quality and might essentially be the make or break when sampling a certain search space. So how would it look to use a meta heuristic in neuronal modeling? The schema is in a way that you start with a model skeleton. So in our case, you choose the formalism like multi-compartment Hodgkin-Huxley, and then you have, for example, the dendrite, you have the diameters, you have the bifurcations. So let M be the equations that describe the cable, as well as the ion channel equation of Hodgkin and Huxley. And sort of ideally, when you come to this point, you were able to fix certain parameters with experimental data, while another set of parameters like this vector p var are now the parameters you didn't constrain with experimental data, and for which you would like to find a good set by using a parameter optimization. So then the search space is essentially the space that's spanned by all these variables, and of course they might not be the full range of real numbers, but it could be, for example, in a certain interval. What you then have to do is you create essentially an initial set of samples of this search space, and then you will evaluate your model on these parameters. So that for each of these parameter sets xi, you're going to evaluate the model equation between a certain start time and end time. You're essentially calculating solving the differential equation in time for the cable equation and the ion channels. Once you've done that, you ideally have some way of expressing whether you like the solution or not. So you calculate a function of the response of the neuron, and that should then allow you to sort that one parameter point is better than another one. So you can create a ranking between the different points you originally started out with sampling. And then, in order to continue, you will retain the better ones, and from there decide where your search continues with a new evaluation. And you keep going until you hit a certain stop criterion. And the stop criterion simply can be you ran out of time, or your absolute value of a metric reached something that is good enough, or you have some other constraints that make you stop the search. So a particular variant of meta-heuristics are so-called evolutionary algorithms, which are population-based meta-heuristics. And they're called evolutionary algorithms because they are inspired by biological evolution. So they use terms like reproduction, mutation, recombination, selection, and they call these points in the search space individuals in a population. So if you look to the right, this is an illustration of how this you can think of it. So you have the first generation, N, which is a pool of individuals, A, B, C, and D. And each individual has a genome which encodes in genes different search parameters. For example, these could be the conductances of a certain type of channel in a certain part of the neuron. With this population, you then generate and evaluate the phenotype. So these channel conductances translate into a neural model, which you can evaluate over time, and then you can create the responses to certain stimuli. And with the mentioned fitness function, a distance function, you might be able to rank and select that sort of you like solution B better than A, better than C, and better than D. And with that information, the evolutionary algorithm can then say, okay, survival of the fittest, solutions B and A were good, so you allow them to, for example, survive into the next generation, and also say well, they're so good that we allow them to mate and create offspring. So you essentially take the two genomes of two solutions and then combine offsprings, combining parts of one channel conductances and other channel conductances from the other neural model. And another element could be, for example, to introduce some randomness by mutating individual channel conductances with a certain probability. The whole idea of this is that this new generation has some knowledge from the previous generation, in the sense that sort of solutions that you ranked good sort of are those that you explore further. So the idea is that you run this mini evolution and hope that this the information about the individuals is guiding you smartly through your search space. By doing all that, of course, the key element is when is a model a good model. So here you have two traces, a red trace and a black trace, and how can you express the difference between these two traces? So one way of it is simply calculating a simple least square distance norm, where you sort of point by point calculate the difference between the two traces. And that will give you an arbitrary unit, a distance of 0.3, for example. When you do that, you have to be aware that that is possibly misleading, because you can find other solutions like the green trace, which has very little to do in terms of having spiking behavior, but in terms of the metric, it will end up having the same arbitrary units distance between the red trace and the black from the red. And this is something which, for the problem of using parameter optimization for neuronal modeling, has been sort of a key challenge. And I think there were several innovations in the last years that sort of addressed that. But before we get there, you need to keep another thing in mind. Sort of these two traces we showed you here, in fact, are the repetitions of the same biological cell. So by applying the same stimulus to the same cell twice, the cell might not react the exact same way. The reason why that is important when you're sort of wanting to guide a computer by helping you find good models is you need to sort of be careful. What is it you're asking for? Are you really asking for that you want to have an exact match of the red trace if the cell is behaving differently the second time you ask it? And therefore, sort of, it might be a useful thing to account for this variability in your search as well. So a method that proved useful, and which this was described by Druckmann in 2007, is that instead of treating every point of a trace equally, you essentially say that maybe what is important for me is that a cell spikes. that these spikes have a certain height of the action potential, that there's a certain time delay to the first spike, and that there are certain intervals between spikes. So instead of saying, want to insist on that this point is correct, you insist you want these basic features of a neuronal trace. And of course, you can choose which features you want and think are important. What these features allow you to do is you can measure them, of course, not only in the model, but you can measure them in the experimental traces, and not only once, but on iterations of the same stimulus protocol. So you can calculate a mean and a variance. So what is the mean action potential height, and what's the variance in the experimental trace? And by then subtracting the experimental mean from the model feature and dividing it by the standard deviation, it allows you to do two things. On the one hand, you penalize the model according to experimental variability. If the experimental traces are very well constrained within a certain distance, you want the model to have the same property. If the experimental trace is variable in a certain feature, you may not sort of care so much whether you hit it exactly or not. And the second aspect of it that comes with it is that suddenly the distance function is no longer in arbitrary units, but it's in the multiples of the standard deviation. So you can say that my model for this feature is within one standard deviation of the experimental variability. So suddenly the distance between your model and the experimental trace is something you can interpret. Another concept that proved to be very useful while trying to advance parameter optimization with neuron modeling is multi-objective optimization. So we just previously described that sort of these different features, action potential height and time to first spike, sort of are different features, but you could now sort of say, I sum it all up in one scalar to sort of guide my search. But instead, multi-objective optimization says you will treat these different features separately. You do not try to sort of trade them in for the other. So obviously you might have many features in your problem. Here we simply depict two for simplicity. And what a multi-objective optimization algorithm does, you're trying to seek for better solutions that might exhibit trade-offs. So if you have different solutions in your search space, you could imagine, again, put here the action potential height and here the time to first spike. So some solutions are very good. They have a low distance value for the model to experiment comparison in time to first spike, but they are not very good in action potential height, while other solutions have a good action potential height, but sort of don't fulfill your criterion in time to first spike. And so if you have different solutions that sort of are landing in different parts of the space, and some of them you can clearly sort out, because essentially there simply you have solutions that are better in all metrics, and therefore sort of you will be able to remove those that are dominated by these solutions that are better in all features. And you keep going until you end up with a set of solutions that are all not dominating each other. So what that means is that these solutions are dominance means that sort of the solution is equal or better in all of its features than the other solution. In this case, these solutions are actually better in some feature, but worse in another feature. So in that sense, there's no clear decision that one is better than the other. It simply means they're showing different trade-offs. And in the terminology of multi-objective optimization, this is called the Pareto form. This is very interesting scientifically, because what it means is while you would like to be here, where sort of you have a perfect match of your model to the experimental expectations, this one shows you that you can have one or the other, and it might prompt you to ask the question whether the search space you've been sampling is the good search space. So maybe you should add another ion channel conductance, or maybe you need another type of time constant in your model. So in this one, this type of using multi-objective optimization for parameter optimization helps you to expose that the search space might not be the right search space in the first place, which you have to always keep in mind when if you model something. It is not a given that the components you put in your model are sufficient to create the behavior you're actually looking for. So putting it all together using an evolutionary algorithm with feature-based metrics and multi-objective optimization proved to be a useful set of mechanisms to advance the automated modeling of neurons. We show that by reverting back to a paper of Matthew Larkham at colleagues, where sort of there's a pyramidal cell which was recorded at different locations with simultaneous patch clamp. And sort of to the right, you see different simulation scenarios. The upper one is where you only stimulate the distal dendritic electrode, and you see some membrane voltage deflection, which is high at the location of the stimulus, lower at the blue, and almost impossible to see in the black one. If, on the other hand, you stimulate the somatic firing with a step current, you'll see the spike generation in the soma, and then the back propagation of the spike to the other electrodes. But then the interesting thing is when you combine the two, so you have the somatic firing in step current injection, and a little bit later, within a certain time window, you do the dendritic stimulation, which mimics a dendritic input, you actually see a nonlinear summation effect that sort of the soma suddenly sends a little burst of spiking activity, and you see a calcium spike in the dendrites. So this has been experimentally observed, and we set out to sort of model this in a model using automated parameter optimization. So this was published by Itai Hai and colleagues, and here you now have a reconstructed morphology. You sort of have the different electrodes set up in an in silico experiment, and you can replay the same type of stimulus protocol, and you see that sort of the model is capable of recreating this non-trivial interaction between two spiking zones of a neuron. So this is a nice demonstration that these parameter search methods in the combination of techniques we showed are useful and create real neuron models. When you do that, you have to consider important pieces. So recreating your experimental expectation, of course, is the first goal of all of that. But at the same time, you have to think further. I mean, it is very easy to get into a scenario where you have overfitting, where essentially your model exactly reproduces the behavior you asked it for, and then fails when you go from a step current to a ramp current. We looked into that and found that actually step currents are quite a good training set, which then have a reasonable generalization when you then, in a second step, test your model on a different stimulus. It's not the same if you trained your model on the ramp current and then went back. So the time constant of very fast time constant and slow relaxation in the step current seems to be useful information for the algorithm to find parameter locations in the search space that then have a more general applicability. So this is an important aspect you always need to consider when doing parameter optimization, that you don't want to overfit and you have to test for generalization. Another aspect is that by design, a meta-heuristic doesn't guarantee that it finds the globally best solution. So it might be finding similar solutions that in your feature space look similar, but sort of the way the value comes about is by very different mechanisms. So especially in these population-based algorithms, you have some solutions which always seem to be very constrained in one parameter, but they show quite a range of expression in other channel conductances, for example. So on the one hand, this could be a problem of the search method, that you're looking in a big search space and essentially you're finding parameters that lead to the same phenotype. But what is interesting about it is that this also should prompt you about thinking whether maybe this non-uniqueness is a property of the biological system in the first place. Because it seems that there's some indication that at least certain types of firing are created in real cells by different channel conductances. So what is therefore interesting when you look at your solutions, you might not only want to pick the best solution, but you want to understand what is the range of solutions and actually ask yourself whether what you're seeing might be even a representation of the biological diversity, but it also could be an artifact of the algorithm. In any case, this values your attention, and for example, Eve Marda and her colleagues, they've been studying this for a long time, and it's useful further reading. Lastly, as with every model, it is important that you understand what the model was built for and what's the range of applicability of your model. So there's an interesting recent review of Almog and Korngreen that took different models, in particular also the model which we showed before, and sort of tested them in questions for which the neural models weren't explicitly built for. So if you do that, of course, you may have good surprises that the model actually is generalizing even into these new contexts, or you have bad surprises that your model actually cannot react and predict properly simply because the information you've been given it when you built the model might not be sufficient. So this you always have to keep in mind, that models have a certain realm of applicability and that you constantly need to ask yourself the question whether you're still within that realm. Let me summarize today's MOOC. So we're in the context of constraining neuronal models with experimental data. Sometimes it's easier than other times to find this experimental data. And for those cases where it's difficult to find the experimental data, what other alternatives do you have? Here we've presented how parameter optimization, in particular meta-heuristics such as evolutionary algorithms, can be used to help you find some of these parameters with the help of a computer. When you do that, you have to help the computer to determine what is a good solution. So you'll have to find smart metrics, such as the presented feature-based metrics. And it is useful to use multi-objective optimization to help you expose certain trade-offs in your parameter space, because it's not a given that the model parameters you're actually starting out with are guaranteed to have a valid solution for the parameter, for the behavior you're looking for. We presented that the different techniques we've shown here can be used to create realistic models, but at the same time you have to always be aware of the caveats. You have to make sure to test that your model generalizes. You have to think about the uniqueness, non-uniqueness of solutions. But overall, and after all, one important thing is parameter optimization doesn't do magic. You have a sample space and you have a certain amount of samples you're doing. So the more free parameters you have, the more difficult it will be for a parameter optimization technique to help you with. So it's always good to look out for more parameters constrained from experimental data and resort to the optimization part as a last resort. Thank you very much for your attention.